Hello students, today we are going to discuss about the topic called as enzymes. Okay, that is the last part of your chapter biomolecules, enzymes, right? So this enzymes, when you are seeing this, enzymes are otherwise called as biocatalyst. So you know what is a catalyst? Catalyst is something that increases the rate of a chemical reaction, isn't it? Means they are having an effect so that if that substance is present, the reaction will be going on very fast. So that sort of substance we call it as what? The catalyst. In the presence of the catalyst, the reaction will be happening at a faster rate compared to that of without catalyst. Okay. So biocatalyst, what of me my biocatalyst? Because since it is taking place, the reaction is taking place inside the living organisms, these enzymes are called as what? The biocatalyst. So if the enzyme is present, then the reaction will be taking place at a faster rate compared to that without the presence of the enzymes. Okay, like for example, for your food digestion, okay, imagine you want to digest the food. This food digestion without the enzyme, it takes a very longer time. Okay, but with the presence of the enzyme, the food will be digested very faster. Means its the rate is very high if the enzymes are present. So that is why they are called as what? The biocatalyst. They increases the rate of what? The biological reaction or the metabolic reaction. And mostly when you are seeing, the enzymes are proteinaceous in forms, means they are made up of proteins. But there are some nucleic acids that are also behaving like enzymes. So those nucleic acid which behaves like an enzyme, like a biocatalyst, increasing the rate of a chemical reaction. So those enzyme we call it as what? The riboenzymes. Okay. Another term what you have to note about the enzyme is that this enzymes, when you see, this enzymes goes and bind to the substrate at particular site. So that particular site where the enzyme is going to get and bind to the substrate, so those sites we call it as, so this is the site where the enzyme is going to get binded to that of the substrate. So this site we call it as what? This site we call it as the active site. So this is the active site of the enzyme where the substrate is going to get binded with the enzymes, and they are going to form enzyme substrate complex. Okay. So this site we call it as what? The active site. So all have understood what is an active site. Active site is the site of an enzyme wherein they goes and binds with the substrate. For the reaction to take place faster, they are going and binding with the substrate. Substrate means what? The reactants. Okay, they are going to bind with the reactants and they will catalyze the reaction faster. So they will make the reaction faster. So a restricted region of an enzyme molecule which binds to the substrate is called as what? The active site. Okay, now some general idea regarding enzymes you should have. Now see, this is a reaction, chemical reaction. Carbon dioxide reacts with water to form carbonic acid. Now listen, this reaction to take place, there is an enzyme involved in us. So what is an enzyme which is catalyzing this reaction, which is increasing the rate of the reaction? The enzyme is carbonic anhydrase. Okay. Now what is the general thing? What they are saying is that in the absence of any enzyme, this reaction is very slow. Imagine it's very slow. It's about with about 200 molecules of carbonic acid being performed in an hour. Okay, so they with if enzymes are not there, only this much molecules will be formed by using the enzyme present within the cytoplasm, that is carbonic anhydrase. If it is present, then what will happen? The reaction will dramatically increase. It will become very fast. Instead of 200, how much molecules you are going to get? You are going to get 600,000 molecules of this carbonic acid can be formed. So here they are saying that the enzyme can accelerate the reaction rate by about how many times? It is 10 million times they can accelerates. It can accelerate any reaction to around 10 million. Not for all reaction it will be the same, but can catalyze the reaction around 10 million times faster compared to that of the absence of enzyme. So enzyme रहेगा तो reactions जल्दी जल्दी होगा. यदि enzyme न रहे तो reaction बहुत ही धीमी से, means it will be going very slow. Okay, in the presence of the enzyme it will be going very faster. Okay, So rate, rate of the reaction means how the reaction is taking place, the speed of the reaction. The rate can be also called as velocity. I can call the rate as a velocity, rate of reaction as the velocity of the reaction if I am specifying a direction. Direction means whether it is moving forward or whether it is moving backward. Okay, the reaction can be like irreversible reaction, reversible reaction. It can go move forward as well as backward, isn't it? So the rate, when you are saying, we can consider it as a velocity of a reaction if I am specifying the direction. So it becomes a vector quantity, isn't it? Scalar quantity, magnitude only, direction is not there, vector, they are having both magnitude as well as direction. So when I specify a direction of a reaction, then instead of rate we can also call it as what? The velocity of the reaction. Okay, the rate of physical and chemical processes are influenced by temperature among other factors. So what they are saying is that many factors are influencing the rate of the physical or the chemical processes in our body. The reactions in our body is controlled by many factors. Among that, temperature is playing also a major role. Okay, so what is they are saying is that a general rule of thumb is the rate doubles or decreases by half for every 10-degree change in either direction. Like for example, if 10-degree rise in temperature in there, then the reaction will become double. It will become faster, it will double up. Okay, 2 times faster it will be moving. But if the temperature is reduced to 10 degrees Celsius, then the reaction rate will reduce by what? Around half the speed. Okay, so half the speed will be reduced if you are decreasing the temperature. If you are increasing the temperature, then it will double up, the speed will double up. In normal pace how it is going, it will be twice that of that it will be moving. Okay, so that is what they are saying. As a general rule, the temperature is playing a major role, and it will increase when the temperature increases 2 times, and it will decrease when the temperature decreases, like around half the speed it will be moving. Okay, and then catalyst reactions proceed at rate vastly higher than that of uncatalyzed. Means if an enzyme is present, if a catalyst is present, then the reaction will be proceeding at a very faster rate than the uncatalyzed reactions. Okay, so that is another fact regarding what? The enzymes. So these are some of the general concepts regarding enzymes. So some of the major characteristics of enzyme that you need to know. Okay, the next topic what we are going to discuss is nature of enzyme action. Very important, nature of enzyme action. Listen students, for this nature of enzyme action with the diagram I will explain, then we will discuss the thing what is given. Okay, so you can find here there is a substrate means what? The reactants. Okay, so this is a reactant. I am adding an enzyme. What is actually this enzyme doing? The enzyme is having, but for each and every reaction there is a particular enzyme. For all substrate, not a one enzyme. So you can find for each and every reaction there are some different types of enzymes. So these enzymes are having some sites. I have told you before, they are called as what? The active sites. So to this active sites, actually what is happening? Of this enzyme, the substrate the enzyme is going and binding to. Okay, so now the active site of the enzyme, the substrate has gone and bind, and what they are producing? They are producing enzyme substrate complex. Okay, so when this enzyme substrate complex, actually what is happening? This enzymes, when these substrates are, the reactants are going and joining with this enzyme, actually making and breaking of bonds are taking place between the molecules. Okay, so as a result of which what is produced? Some recombination and all taking place, this bonds are broken, and chemicals are mixing up, and they are producing a new product. So what are produced? New products are produced and they are released. So the products are released, and after that what will happen to the enzyme? The enzyme will come back to the normal position. Means they are normal, they are not means they are not getting involved, and they are also not producing new products. Okay, the enzyme will be safe, that will be separate. Again this enzyme can go and attack with that of the substrate, and the reaction can be again produced. They will release the product, and again they can go and attack with the substrate. Okay, so like that the reaction will be taking place. So it's hope so it's clear. So enzymes, when they react with the substrate, they form a bond between them, that one we call it as the enzyme substrate complex is formed. This enzyme substrate complex will act as a catalyst. If this is not formed, for the substrate to break down, it will take a longer time. But this enzyme substrate complex, when it is formed, the bonding as well as the breaking and all will be taking place very fastly, and the product will be released and the enzyme will be separate. Okay. So, enzyme as well as the product will be released. The enzyme can again go and bind with the substrate and the reaction can again proceed. Okay. So, like this the are acting as what? The enzymes are acting, and this is how the enzyme action is taking place. So, now I will just read out. A catalytic cycle of an enzyme action can be described in following steps. First, the substrate binds to the active site of the enzyme, fitting into the active site. Yes, they have gone and fit into the active site. Next, what is happening? The binding of the substrate induces the enzyme to alter its shape. Now, what happens? The enzyme will to some extent alter the shape, fitting more tightly, and this active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond of the substrate. So, what is they are doing? I told you, they are breaking the chemical bond which is present in the substrate, and they are producing a structure called as what? Enzyme product complex is formed. Okay, so after breaking, enzyme product complex will be formed, and this enzyme releases the product of the reaction. Thereafter, they are releasing the product of the reaction, and the enzymes are becoming free, is ready to bind to another molecule of the substrate and run the catalytic cycle once again. Understood? So, what are they doing? They are going and first binding with that of the active site of the substrate. So, enzyme active site is going and binding with that of the substrate. They are forming enzyme substrate complex. Now, more fitting will be taking place. What will happen? The bonds and all will be broken. Then the bonds and all are broken. They are forming what? Enzyme product complex. After that one, what is happening? The products are released. The enzyme is released. The enzymes is again going and continuing its cycle. Okay. So, this is how an enzyme action is taking place. Hope it is clear.
Next, we are going to discuss about what do you mean by the term activation energy. Okay, so what is activation energy? Enzyme function, in that only activation energy. Listen students, so the reactants are reacting and a product is formed. So if an enzyme is present, the reaction will be going on at a faster rate. If an enzyme is absent, the reaction will be going in a very slow rate. That's what we have seen, isn't it? So why is with the presence of enzyme, the reaction is going at a faster rate? Okay. Now, for this, we are going to introduce a concept called as activation energy with the help of a graph. Okay. So this is a graph. On the y-axis, you can find the potential energy. Okay. The energy which is required for the reaction. Okay. And you can find this one when you are seeing the reaction, how it is taking place. Okay. So this is the rate of the reaction, progress of the reaction, which is going on, progress of the reaction, and here when you are seeing it is the potential energy of the y-axis. X-axis it is progress of the reaction. Now listen carefully what is happening. This is the substrate. S is the substrate which is present over here. Okay. This substrate when you are seeing it is getting converted into a product. Okay. So the reactants are getting converted into a product in a reaction. Now, when this reactant is getting converted into a product, actually what is happening when you see, there are two types of reaction you have learned. One is exothermic reaction and another one is endothermic reaction. The reaction which gives out energy, releases energy, we call it as what? Exothermic. And the reaction which absorbs energy, we call it as what? The endothermic reaction. So, one which gives out the energy. Okay, so they are releasing the energy. You can find the substrate is product, means the energy is released. Okay, so automatically the energy, potential energy will be lost. It's gone. Okay, so the substrate will be producing the product somewhat here down because the potential energy is less because the energy is being released in that reaction. Okay. And another one is that endothermic. In this, when you are saying the substrate is absorbing the energy and the product will be formed somewhere here. Okay, so somewhere here only what is produced, the product is produced. Is that clear? So, substrate when it is forming the product, when it is exothermic, the product will be somewhat formed at the bottom part, And if it is endothermic, the energy is absorbed and the product will be formed at somewhat above this thing. Okay, above the x-axis, the product will be formed because it is an endothermic reaction wherein they are taking the energy in. Exothermic, they are releasing the energy. Okay. Now, if an enzyme is present and if an enzyme is not present, actually, how is this reaction going to take place? Now, the substrate, if an enzyme is not present, they have to undergo a huge transition state. So, transition state means from the substrate to the product form. The state, when you are seeing the reactions which is taking place, slowly they are getting changed into the product. That transition stage is a bit bigger, isn't it? The big height, when you are seeing, it's much height is much good, means you are having a lot of height. But if an enzyme is present, then you can find what is happening. The thing is, it is very less. The energy, activation energy is very less for this transition state. Listen, so this is the transition state. So when there is no enzyme, okay, so they have to spend a lot of energy to form a product. So you can find how much amount of energy they are going to spend from here to here. This much amount of energy is needed. That energy for activating the substrate to form a product, that energy we call it as what? The activation energy. This is the activation energy which is needed without an enzyme. Okay. So you can find they have to, a hill is much height, it is having much higher in form, isn't it? Now, when an enzyme is present, so when an enzyme is present, I have told you, imagine this is the enzyme and this one is the active site. Okay, so the substrate is going and binding with this active site, isn't it? So what is actually happening? When the substrate is going and binding with the active site, there is an energy between these complex form. That energy we call it as the binding energy. What do we call? The binding energy. And what does this binding energy do? This binding energy helps in breaking down of the bonds between them very easily, and thereafter they do not cross the big hill. Immediately you can find fastly they can release the product. Okay, so there when the enzyme is present, the activation energy which is needed is less. So activation energy with the presence of enzyme is very less, but activation energy without the presence of enzyme, it is much higher. Like for example, I will use an example and let you know. Now for example, I am going to carry out a task myself. Okay, then I have to put in a lot of effort. Okay, so after that putting in a lot of effort, I am getting the product. That is the result. Okay. Now, if imagine I am having someone as my backup. Backup means what? Who is helping me out. Okay, my partner is there and he is very efficient enough. He or she is very efficient enough. In that case, what is happening is that I need not put that much effort. Half of the work is done by him because he is very efficient enough. Okay, so what he will do? He will come and bind with me. He will help me out. So when he is helping me out, what happens? Already he is having that potential within him, that energy within him. Then whether I have to spend that much energy like that? No. Like I need to spend only 30% or 20% is enough because already 70% he is highly efficient. So what will happen? The work will be carried out very faster when one person is there. The same thing here when the person is the enzyme, and that enzyme is having actually what, when the enzyme and the substrate both are reacting together, they are forming what? A binding energy is present between them. This binding energy is capable enough to make out the process much faster. The breaking down of the bond and mixing will be much faster and the product will be obtained very easily. Understood? So activation energy without enzyme is much higher. You have to climb a lot. You have to put in a much effort to get the product. Okay. But when the enzyme is present, you can find the activation energy is very less. You can easily cross and the effort is very less and the reaction rate will be much faster. Understood? So that is the thing regarding what? The activation energy, and this stage we call it as what? From the substrate to the product forming, that state we call it as what? The transition state. For this transition state, activation energy is needed. For the activation energy, without the enzyme, it is much more compared to that of the with enzymes, it is very less. Hope it is clear.
Factors affecting enzyme activity. The first factor is your temperature and pH. Listen students, actually there is a condition called as optimum. Optimum means this much temperature, like for example in human beings it is 37 degree Celsius. Okay, so that is the optimum temperature, like that optimum pH. So for some enzymes it should be acidic, for some enzyme it will be basic, like that. There is an optimum pH for each and every enzyme, an optimum temperature for each and every enzyme in order to function very effectively. So below that or above that means below that optimum temperature or below that optimum pH and above that optimum temperature or above that optimum pH, the enzyme activity will be very less. Okay, it will gradually go down below that optimum, and above that optimum gradually what will happen, it will be going down. So in a graph if you are representing, you can find so the enzyme activity you can find as the pH is going on increasing. So what is happening, the enzyme activity is going on increasing, and it has at an optimum condition, optimum pH, the enzyme activity is at highest. Thereafter, if at all the pH is going on increasing, whether the enzyme activity will increase? No, it will be gradually coming down, which means there is an optimum pH at which the enzyme activity will be highest. Below that or above that, when you are seeing the enzyme activity becoming lesser. Okay, so like that for temperature also, there is an optimum temperature at which the enzyme activity is highest. Below that as well as above that, when you are seeing, the thing will be very less. Like for example, if I am increasing the temperature, the enzyme activity goes on increasing, but up to a limit. That optimum, when it is reached, thereafter if you are increasing the temperature, what will happen? The reaction will be moving fast? No, the reaction will be going down. Okay, so that you have to make a note, that is the temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows highest activity at a particular temperature and pH, called as what? The optimum temperature and pH. Activity declines both below and above the optimum value. It is going to reduce, it get reduced. Okay, low temperature, what happens, preserves the enzyme in the inactive form. So when you are keeping any food materials or anything, why you are keeping it in the fridge? So that the food is not getting wasted. Why? Actually under that low temperature, the enzymes which are present in the food materials and all are not reacting. Why they are not reacting? Because they are proteins, they are becoming inactive under low temperature. Okay, and when you are increasing the temperature, what will happen? The enzymatic activity is destroyed. Why? The protein, when you are seeing, it is denatured, it's becoming functionless. Okay, so denaturing is taking place, the bond and all is broken. So automatically what will happen? The enzyme will not function. So below also it is inactive, and above the proteins are getting denatured by heat. So the enzyme's activity will be very less. So there should be an optimum at which the enzyme activity will be more. The second thing what they are saying is that the concentration of the substrate also determines the rate of the reaction. Okay, so concentration of the substrate. Concentration of the substrate means what? The reactants. When the reactants are available in less amount, the active means enzymes are binding with them, and the activity is going on. When the substrate concentration is going on increasing slowly, when you are seeing the substrate concentration is going on increasing, that is the reactant concentration is going on increasing, then what will happen? More enzymes will be going and binding with that of the substrate, means reactants which are present, and the reaction will be going on at a very higher rate, isn't it? So gradually you can find they are increasing slowly the reaction is going on increasing. When the substrate it is increasing, the reaction is, velocity of the reaction is going on increasing when the substrate increases. But what will happen after some time? After some time, what happens is that the enzymes which and all were present has all gone and bound with that of the substrate. Now more substrate if you are adding, what will happen? Whether the enzymes will be present there? No, the enzymes will not be present. Okay, immediately you cannot find the product is formed. It will take a bit of time, isn't it? So instead of giving them time, the enzyme to release and come back and bind to the substrate, If you are going on increasing the substrate, then what will happen? The substrate will not be binded by the enzyme. Gradually you can find there is a drop, okay, drop in the velocity of the reaction. So that's what they are saying. When you increase the concentration of the substrate, to some extent what happens, the reaction at the first stage it will be going very fast, but after it is reaching the velocity, maximum velocity, okay, thereafter you can find, if you are adding the substrate more also, enzymes are not there, enzymes are very fewer to carry out the reaction, so automatically the reaction rate will be coming down. Okay, so I will read out. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The velocity ultimately reaches a maximum which is not exceeded by any further in concentration of the substrate. So if you are adding more substrate, the increase is not taking place. This is because the enzyme molecules are fewer than the substrate molecules. What happens? The enzymes are fewer, less than that of the substrate molecule, and after saturation of these molecules, there is no free enzyme molecules to bind with the additional substrate. So with the additional substrate to bind, there is no enzyme molecule present, so that the substrate, they will go and bind the reaction to continue. Understood? So these are the factors which determines the rate of the chemical reaction. Okay, so one is your temperature. Temperature, we told you, heat energy causes mole collision between the enzymes and the substrate. So optimum when it is coming to 37 degrees Celsius, the temperature is very optimum for the reaction to go at a very high fast rate. Thereafter what will happen? The enzyme is denatured at the high temperature. So for pH and temperature, there should be an optimum pH and optimum temperature below and above which reaction will be very less. Okay, substrate also gradually it increases after reaching the maximum. Since you cannot find the enzymes are available, the reaction decreases. Okay, so that is what they are saying. So you are saying when first it is increasing, isn't it? So first it is going on increasing, the reaction is going on increasing, but after reaching some time, there is no enzyme molecules to bind with the substrate, so then the reaction will decrease. Okay, with this I will stop for today. Our next part we will be discussing it in the next class. Thank you.